Hi, everybody. My name is Carol Hamilton. I am the Vice President of Development here at the ALS Therapy Development Institute, and welcome to the Town Hall, offering you a live demonstration of the ALS Research Collaborative ARC. We are grateful to have you here, and we're excited for you to get to see this. I want to make sure we thank our sponsors who help us throughout the year put on our educational programs. Thank you to MT Pharma and Cytokinetics, Biogen, InSource Services, and Apellis. We are very, very grateful. I want to also introduce some of our development team who will be contacting you after this town hall to get your feedback and to say hello and to answer any outstanding questions you might have. And I know they will be both outstanding and potentially outstanding. So Megan Mania, Glynis Murray, Bill Johnston, and Michelle Dupree. Yes, wave your arms around if you could. You will hear from these people afterwards. We are going to keep you on mute during the presentation, but we will have plenty of questions at the end. So bear with us for that. And I'm now going to introduce our Manager of Science Communications, John Gang, who will provide a brief overview of ALS TDI before we hand it over to Dr. Vera.
Hello, everybody. Once again, I just want to welcome you to today's ALS Town Hall on the ALS Research Collaborative. And once again, thank our sponsors whose logos you can see at the bottom of the screen there. As Carol said, my name is John Gang. I'm the Manager of Science Communications here. And you might recognize my face from our blog or my voice from our podcast, the Endpoints Podcast, which is available on Apple Podcasts and Spotify. If you are not subscribed, there's some good content on there. I suggest you go seek it out. And also, as Carol said, we're just going to run through a quick introduction to ALS TDI. I will keep it brief because I know a lot of you on here have heard this spiel before. And then we will hear from Dr. Fernando Vieira, our CEO and Chief Scientific Officer, for a live demonstration of the ALS Research Collaborative. And then we will have some space for your questions. And for those of you who are not familiar with ALS TDI, our mission is to discover and develop treatments for everyone with ALS. And we were the world's first nonprofit biotech. We were founded in 1999 by the family of Stephen Haywood. Down there in the left-hand corner of the screen, you can see a picture of our first office, which was in the Haywood's basement in Newton, Massachusetts in 1999. And in the orange there is Stephen Haywood. He was diagnosed with ALS at the age of 29. And when his family found out that there were no available treatments, they decided to do something about that and started this organization. And today we are the most comprehensive drug discovery lab in the world focused solely on ALS. We have a state-of-the-art laboratory in Watertown, Massachusetts, and we continue to put 87 cents of every dollar we raise into finding effective treatments and cures for ALS. And down there in the right-hand picture, you can see Stephen's son in the blue shirt with some of our scientists in our lab. And unfortunately, Stephen passed away in 2006, but his legacy and his family's mission is still central to everything we do here. And ALS TDI aims to be the drug discovery engine for ALS. And what this means is that we do the rigorous research that is needed to fill the ALS clinical pipeline with potential treatments. That means we work to discover and invent ALS drugs in our own lab, and then when we find a promising one, we partner with other companies to help ensure that drugs advance to trial rather than bringing it to trial ourselves like most other biotech companies. And we do this because we do not want to follow one drug. We want to go back to the drawing board so that we can continue to do this over and over again, because we know that ALS is a complex disease and is going to take many treatments to finally end it. And with this in mind, we will not stop until everyone with ALS has access to effective treatments. And over the past 2+ decades, our impact has been rigorously testing more than 500 drugs in an animal model of ALS, which is more than any other ALS research lab in the world. We were the first nonprofit biotech to invent a potential ALS treatment called tegoprubart and bring it from our own lab through FDA review and into clinical trial. And that drug did complete a phase 2A study last year. And we founded the first ALS Precision Medicine Program, or PMP, which was a partnership with people living with ALS all around the world to gather data about the disease and speed up drug discovery. And now that program has evolved into what is now known as the ALS Research Collaborative, which is a global initiative to accelerate ALS research by continuing to gather data and sharing this data with the ALS research community. And with that, I will hand things over to Dr. Fernando Vieira to tell you more about the ALS Research Collaborative.
Thank you, John, and thank you everybody for being here. I'm always amazed at how many folks find the time and invest the time in their day to join these town halls. Thank you so much for your interest. So, like John said, I'm going to talk a little bit today about our ALS Research Collaborative. I'm really focusing today specifically on the ALS Research Collaborative Data Commons, and I'll get into what I mean by that. First, you're stuck with a few slides before I get into the basic demo of the program. So, like John said, ALS Research Collaborative is really an evolution of what we had referred to for a long time as the Precision Medicine Program. The Precision Medicine Program was initiated in 2014, and it was really the result of the prescience of my predecessor, Steve Perrin, and our overall scientific leadership at the time, to recognize that ALS is a really complex disorder. And in order to make a dent in this disease, we're going to have to really learn a lot about the disease directly from people with the disease. And so there was an effort to create a program that would enroll people with ALS directly into the program. We didn't have clinics involved. This was sort of direct to a person in their home. And we'd be collecting information about their disease presentation, their background, family history. And we'd also, along with those types of data, we would collect new types of data that hadn't been collected yet, so digital biomarker type data. So accelerometers were implemented, voice recordings started to be collected. These were things nobody was really thinking about in 2014. And then on top of all of that, we integrated whole genome sequencing and cell biology. So it was really a catch-all program to learn as much as we could in a connected way. It was a highly dimensional program. So lots of different dimensions of data from as many people as we could. And through those intervening 8+ years, we ended up, thanks to the generosity of time and effort of so many people with ALS, enrolling more than 850 people in that program, some of whom have been in the program since. They've been fortunate enough to live a long time with ALS, and they've been contributing data almost monthly during that entire time. So it's this massive data set where we collected a lot of information. Now, through the ALS Research Collaborative, we're emphasizing the data sharing element of this program. We've always partnered directly with people with ALS by sharing the data that they've contributed to us back to them through their patient portal. But now we're expanding that to really aggregate the data and share it with other researchers in the world, from academia, clinics, and industry, to really unlock the power of it all. So I'll walk through a little bit of that with you. So the goals of ARC are aligned directly with the goals of the Precision Medicine Program, or PMP, through the years. Number one, we're trying to discover new therapeutic targets. ALS TDI, we consider ourselves a drug discovery engine for ALS. In order to effectively discover therapeutics, we need to find the best ways to target those therapeutics. And we need to find the right people in whom to target those therapeutics. And that's a major emphasis of ARC, this program. We also, like I mentioned before, would like to establish better biomarkers of disease progression. You've probably all heard about the ALS functional rating scale, or many of you have likely heard about the ALS functional rating scale, a survey essentially that can track disease progression. We believe there are other ways that may be complementary or better. And so we've implemented accelerometers and voice recordings and machine learning to see if we can extract a better signal. If you can measure disease progression better, you may also then be able to measure the beneficial effects of therapeutics better and more efficiently. So that is another big emphasis of this program. We've also been working hard to identify or validate blood-based biomarkers. In 2019, as part of the precision medicine program, we implemented an in-home blood collection program where we send phlebotomists to people's homes to collect blood samples from folks roughly every 3 months or quarterly. And the idea there is to delve deeply into those samples at an omics level. You've probably heard of genomics. Well, there's also transcriptomics and proteomics and metabolomics, et cetera. These are all the study of the big data, the many, many things that can be measured in a biological sample. We're doing that in these blood samples to see what relates to ALS, what relates to faster disease progression, what might relate to slower disease progression, so that we can measure the disease that way as well. And then finally, and a big emphasis here, like I said, we've always shared data back with our participants, but now we're sharing the data in aggregate back with the ALS patient community, hence the ALS Research Collaborative. Just to give you a little bit of a sense of where we're going with all of this next. We've been working very hard to enhance the participant experience. So over the past year or so, our clinical operations team, led by Alan Primaseri, our communications folks, led by Megan Lawler, and our IT team, led by Sean Sullivan, have been working really, really hard to improve the sign-up portal for new participants and to upgrade the participant portal where they can access and contribute data. And we're going to continue to enhance that over the next few months and years. Another major objective is to increase our total enrollment to 2,000 people with ALS. Over the past 8+ years, we've been able to enroll 850. It's great, but it's not enough. And so we recognize that. So we're working with our institutional review board to get approval to expand the patient population. And when we do, we're going to be aggressively going into the community and asking folks to contribute. We're also working right now, and again, pending institutional review board approval. And if you don't know what an IRB is, or an institutional review board, an IRB is the organization that oversees all clinical research that we or other groups might be doing, and makes sure that we're acting appropriately and we're treating our patients, our participants appropriately, and keeping data secure, et cetera, and keeping them safe. So that's why we have to go through an IRB for approval. But in this case, we're also looking for approval to integrate electronic health records. So any participant who consents into our program would then have an opportunity to consent a second time through a separate portal to integrate elements of their electronic health records that they think could be helpful to ALS research. It's not required in any way for a participant, but we're looking forward to being able to add those data as well. We intend to continue to add new data types into the data commons. What I'm going to show you today, the platform where we have all of the data that researchers can access or community members can access. We're going to add new data types to it over time. And then finally, we want to work with external groups to implement a global unique identifier. So that would mean a tie-in through an anonymized identification number so that we could connect easily to other research programs. And I've had conversations with other folks in the ALS space who are working on this and really look forward to implementing this to kind of, one more time, have a force multiplier effect of all of these data. So what I'm going to walk through today is the ARC Data Commons. Like I mentioned, I'm calling this a beta launch because there are still some kinks and there are still things that we're going to work out. And anybody who's getting access to the data commons right now, signing up in spring of 2023, we're looking for your feedback on how to make it better. This is not yet a finished product, but I think it's really good. The program leverages right now about 35 terabytes or a lot gobs of data that we've collected through the ALS Precision Medicine Program, now ARC. We do have the sharing of the data is approved by our institutional review board, and we're sharing it with nonprofit organizations, academics, and industry researchers. And we're absorbing all of the costs associated with cloud computing and queries for nonprofits and academics. And I'll show you next that it's kind of a unique set of dashboards that are really designed to facilitate really quick questions and getting quick answers for researchers. And this isn't limited to researchers. If you're a person with ALS who has a really a keen interest in the biology of ALS and wants to dive into this, you have access to. So you'll be able to submit an inquiry and get access as well. And also one other point, if you are a person who signed up for access and while you're there, you can also download the data. You're not limited to access to the data while you're logged in. You can download the data locally to your own machine and use your own local software tools for your own statistical analyses as well. So the data use agreement might last a year, but you still have the data long after that. And with that, I'm going to shift over into demo mode, which I'm going to admit is a little bit terrifying because you never know how live demos are going to go.
And Fernando, as you're doing that, I just wanted to help make clear that you were saying that a data use agreement might last a year, but you would expect that people might renew for
renew. Yeah. Yeah. We fully expect for folks to renew. Absolutely. Thank you for pointing that out. So you're now looking at the ALS Research Collaborative Data Commons, or ARC Data Commons dashboards. There are multiple dashes that we developed. And when we were creating this, we asked, what would we want to be able to have access to? And by we're not all at ALS TDI bioinformaticists or biostatisticians. We're bench scientists and clinicians mostly. And so we asked ourselves, what might bench scientists and clinicians want to ask of this data set, and how do we make it easy for them to do so? And so that's what we ended up building. It also works great if you are an informaticist or biostatistician, because you can download all of the tables you could ever want to your heart's content and run it through RScript if you want. But if you're not that, this has a low barrier to entry, and I'd like to demonstrate that. I'm going to show you 3 different dashboards today. The first one I'm going to show is the ARC cohort viewer, which allows you to really delve into some of the key elements about the folks who have been contributing data over time in this program. Okay, so first, the main feature of this dashboard is that you can slice and dice and filter the data any way you could imagine in order to look at specific subtypes of people with ALS. So the first thing I'm going to do is show you what the entire cohort looks like, what everybody in our program. So people who have signed up. So you have all of these different filter buttons here: participant ID, country, region, etc., site of onset, age of onset, all of these different features. I'm going to say sign up anytime. I'm going to load the anytime cohort. I have it set. We do have healthy controls in this. I'm going to filter those folks out. I'm going to have only people with ALS next. And so we're loading data into each one of these tiles that tell you a little bit about our cohort. So we've had more than 1,100 folks who have consented. A person who has consented is not considered fully enrolled unless they have contributed at least 3 months worth of data. A main element of this program was that we wanted to capture data over time. So that's why those criteria are set. So we have what you're looking at, a number of folks who have given us those 3 data points. Folks who have this value here, this 7,000 value, represents the number of weeks worth of accelerometer data people in our program have contributed. So for 1 week out of every month, we send to many of our participants 4 accelerometer devices that they can wear on each of their wrists and each of their ankles. And we collect those data. That's what this represents, is each unit is 1 week worth. We have almost 10,000 voice recordings. We have whole genome sequencing data from a lot of these folks. These are blood draws from folks directly from their homes, etc. You can see our distributions here. We have a slight enrichment for men versus women, but that's not that shocking given what we know about ALS demographics. As we scroll on down, we have different tables. All of these tables are completely downloadable, but here are basic demographic information about each of our participants, the number of data points they may have contributed. So here we have an individual who contributed 27 ALS functional rating scale data points. So that's 27 months worth of FRS data from that person, 14 months of weekly accelerometer data, etc. Going on down, these are ALS-associated mutations. I'll delve into that more deeply through a different dashboard. I'll show you that later. We also have repeat expansion analyses. So obviously what most folks focus on when it comes to ALS is C9orf72 repeat expansions, but we're also looking at other repeat expansions as well. Ataxin-2 has been implicated. Some of these haven't been implicated in ALS, but they've been implicated in neurodegenerative diseases, and we think they could be important. So we're delving into that as well. Here we have some graphs representing spaghetti plots, is what these are often called. So ALS functional rating scale of individual people over time. This will be more useful once I start filtering into different subsets of people with ALS. I'll show you that in a moment. And here at the bottom, it's important. This is a histogram. So if you're not used to looking at a histogram, we're looking at a histogram of progression rates. So on the left, most categories here are bins of people who have or bins of data that are from people who have ALS progression rates that are quite fast. So, you know, -6, we have 2 people who are losing about 4.5 FRS points per month. That's incredibly fast. Most of our folks are slower progressors, much slower than what you would typically see in clinical trial databases. So if you looked in the PROACT database, where most of that is data from people who are in placebo-controlled cohorts of clinical trials, they tend to be losing about 1 point per month. That would be around here. Most of our folks are closer to losing 0.2 or 0.4 points per month. The reason for that is we invite any person with ALS to join our program at any time in their progression. And it doesn't matter if they're fast or slow. It means that people who are slower progressors have more time to find us. And so we welcome them, but it does mean that there's an enrichment for slower progressors in our cohort here. So now that you've seen what the overall cohort looks like, one could ask lots of questions about specific types of profiles of folks within this cohort. So, for example, if somebody wanted to isolate only people with, say, onset affecting their bulbar onset, let's say bulbar onset, one could go through each of these strings here represents any assortment of onset that has been reported to us by our participants. So I could select, I would go through and look for features that are related to bulbar onset. I can select all of them, one at a time, swallowing, tongue, swallowing, swallowing, tongue. So I'm looking for folks with bulbar onset, even if they had other symptom types as part of their onset as well. You could do this any way you'd like. I might miss one or two during the live demo here, so I apologize if I do. You can call me out if I do in the chat. You can see there are a lot of different versions of onset types reported in our program. I'll also point out while I'm doing this that these data are updated daily. So a new participant might join this week, and they may have a different profile of onset type that would end up being reflected here, maybe not the next day, but within a week, essentially. All right, so now I'm going to filter on those criteria, and we'll see how many folks we have. So we had 265 people in our cohort who reported among their first symptoms were bulbar symptoms. Slightly less enrichment for men in this subcohort than women, but still more. This is now the basic profile of this subset of folks. These are the spaghetti plots of these people now. And now when I go down to the histogram, actually, I hadn't done this before. You see that things are a little bit left shifted, right? So the peaks are not so enriched for people who are slower. You see higher peaks now happening in the faster progressing, which isn't shocking based on what we know about ALS, that bulbar onset tends to correlate with faster disease progression. But you can instantly get this answer. This is the type of question that frankly, before we built this tool, would have been quite hard for me to ask of our own data set. Now it's very, very easy to ask, and you could export all of the data associated with these people. And if you wanted to, you could export all of the single nucleotide polymorphisms that have been associated with ALS that are within this subset of people. So it's really powerful to be able to link the phenotype to the genotype data that we have. I hope you're following along. I know some of the words I'm saying, so gosh, I get caught up in myself. Genotype. So phenotype, that's the way you present clinically, the physical manifestation of the symptoms, right? A genotype is what are the genes that you have, right? What are the genetic polymorphisms or lack thereof that you have? I hope that made it a little bit clearer. So there are literally thousands of ways one could try to filter these data and isolate specific subsets of people with ALS. The next way that we find that we often end up doing it is to focus first on the genetics. So what I showed you here is a focus on symptoms. Next, I'm going to focus on specific genetics. So I'll go into our list of dashboards, which we have. So genomic dashboard, we're currently again in the cohort viewer. You're seeing cohorts. Now we'll go to the ARC genomic dashboard. And so we have different filters at the top this time. So here, one could select if you wanted to look at a single point mutation and find a cohort of people who have any polymorphisms in any of these genes, many of which have only been tentatively linked with ALS. This is a very inclusive list of genes.
Fernando, could you define polymorphism for people?
It's like a change in the spelling of a word in your genome almost. So that's a way I think about mutations are misspellings in a gene, right? Or kind of a misspelling. A polymorphism can be a mutation, but isn't always a mutation. You can have a change in the spelling, but by one definition, it's still correct. It's just different. And so a polymorphism includes things that are mutations and just a little bit different. Does that make sense? Did that answer it? So not everything here is considered a mutation, but they are considered a variation, I guess. So it's a long list. Like some of them might be very familiar to you. You're going to see your SOD1. You might see another one that's quite familiar to people is probably, your TDP43. It's TAR DNA binding protein, TDP43. Another might be you obviously C9orf72. So I'll actually pull the C9 data if you're interested, but you can go through any of these and isolate folks who might have a mutation. So I'll do a relatively rare, tentatively implicated gene that I think about a little bit. It's an RNA binding protein, EWSR1. And let's see. Pretty instantaneously, we can find our participants who have polymorphisms in this gene. We can see they're relatively rare polymorphisms, so they're only showing up in about 2 out of 1,000 people. It's considered uncertain significance, but if I wanted, it has been associated though with ALS tentatively. And if I wanted to look at these folks, it would be really easy for me to do that. So I just grab these, I copy, I go back to our cohort viewer. I make sure it's set to sign up for any time. I paste those IDs in, I update. And so now we're finding the 11 people who have contributed 87 weeks worth of accelerometer data and 72 voice recordings. Pretty typical proportion of men to women. We actually have, and a reversal. So this is part of Rick Bedlack's reversal cohort, who he's shared with our program. And we have healthy volunteers who may have a family relation. That's often the case. If I wanted to filter those folks out, so I'm just focused on people with diagnosed ALS, I can do that. It's a pretty responsive program. And so these are the ALS functional rating scale curves for each of these individuals. And this is the histogram of their slopes. These folks, there's a wide variety, but they are definitely more enriched for FAST than the overall cohort. Now, one of the really important things to consider is that these people don't only have these mutations. It's very, very easy to overinterpret. If you go hunting for your gene of interest and you find those folks, to be a bit myopic about it and think, oh, their disease is attributable to the gene that I care about right now, the one I'm thinking of. But the reality is much more complex than that. And this dashboard makes it easy to notice that it's much more complex than that. You can see the other polymorphisms that have been implicated in ALS, even tentatively, that are present in each of these people, right? So here's participant 62. I tend to sort these by rarity because some of these are quite common, and so you wouldn't think they're very clearly implicated in ALS. But we have a person here who also has a rare ataxin 2 mutation, another one who has a rare ataxin 2 mutation. Now, this makes you start another person who has an alsin mutation, an ALS2 mutation. So it forces us as researchers to confront the complexity and recognize that there's a lot more going on, but maybe also to look at relationships that we might not have stumbled across. Is there a potential relationship between EWS15 and ataxin 2? Maybe that's the rabbit hole one might go down now and want to explore in a separate cohort from another database. Ask the same question from a separate cohort. There's a lot of power in that. I hope you guys are following along okay. I want to go back to the genomic dashboard to show how we can do the same thing with repeat expansion mutations like C9orf72. So we also have C9 repeat data associated. I set the gene name for the repeat expansion. I set is pathogenic. And then when I update, now we're pulling from the cohort, we're finding the folks who harbor repeat expansions. Each of these people have we have 2 copies of most genes in our genome. The first copy in each of these people is considered normal with repeat counts of 5, 2, 8, 2, etc. Their second copy with repeats much larger. So considered in ALS literature, there's a general consensus that greater than 30 repeats is pathogenic. Some people have said greater than 24. Each of these individuals harbors a repeat longer than 30, and so defined as pathogenic. If I wanted to then look at this sub-cohort of folks, I again copy their IDs, go back to the cohort viewer, paste these in, set it again to ALS patient, make sure that the sign up is any year, update. And now we have our new cohort. These folks, 300 weeks of accelerometer data, 327 voice recordings, etc. Not all of them reported a family history of ALS, meaning some of these people were sporadic ALS patients as diagnosed, but did end up harboring the C9 mutation, which is an important distinction. Here we have less enrichment, a little bit more enrichment for women. I'm not saying that means anything. It's just I'm pointing out that you can see it quickly. And what we see here for C9, there's at least in our cohort, we have wild variability in clinical presentation, with some people having very slow progression and some people having much faster progression. And this is all people harboring the same or similar mutations. So there's a lot of power, though, in being able to quickly look at these data. If I wanted to understand more about one specific person, you can then do that. You can go into the participant overview. So here we have participant, the identifier here is 383. So I can go into a different dashboard. This is now the third dashboard that I'll be showing you. And then I can update. And here now you have a breakdown of the FRS scores that this person has contributed over time while they've been in our program within each category. These graphs are anchored to the time when they reported their onset. So they might have joined our program at approximately a year into their disease, but if they reported their onset of symptoms a year prior, that's what ends up being the anchor point for these graphs. And you can see here we have a person who, when they came into the program, already had serious impairment of their ability to walk, less impaired ability to cut food, etc. Now I can scroll further down. Here's their overall FRS scores. And these are their accelerometer data, right? So this person here, what you're looking at are data representing their right ankle. That's what the RA, average normalized, means, right ankle, left ankle. This is the orange line represents a core function. It's right wrist, left wrist. What that represents is a person's ability to do a seated crunch and then back. And then you see right wrist and left wrist. So this is pretty consistent with their FRS data. They came in reporting that they were having trouble climbing stairs. They were an FRS score of 1. Their accelerometer scores were lower for their legs than for their arms. And let's see, did this person contribute as well? This person also contributed voice recordings. These data are interesting and quite unique. So about a year ago, we published with our collaborators at Google an algorithm where we trained a machine learning algorithm to recognize bulbar disease severity by listening to a voice recording. And so here we have what may be a relatively objective measure of this person's bulbar disease progression over time. I haven't seen this person's data actually until now. And then this individual's ALS-associated polymorphisms are tabled specifically for them here. This individual's repeat analysis. So like we said, here you'll find again, if I look, I'll find the C9. Where was it? There it is. So we'll find their first allele had just 2 repeats, normal. Their second had 76 repeats, and that's pathogenic. And this person, sorry, I have to move the screen, reported these medications that they were taking these, and they reported when they stopped taking these medications as well. So there's a wealth of information that one could have access to through this tool that really lets you have a complete picture at both a cohort level or at an individual level about the disease biology. And again, as we continue to collect more and more data types, so as we generate RNA-seq data, so measuring many, many RNAs out of a blood sample, or proteomics data, again, measuring many different proteins out of a blood sample, we intend to integrate these data over time, along with the whole genome sequencing, along with the clinical outcome data, that can be really, really powerful for us and for others to discover interesting targets and to better understand this incredibly complex disease. And with that, I will open it for any questions.
So Fernando, I am sensing from the chat, I don't know if you've been able to see any of it as you are presenting, but clearly there is a lot of enthusiasm, particularly around the researchers who are on the call. So thank you, everybody. I want to make sure too that those that are not researchers are understanding how this will be real world applicable and could potentially speed therapeutics. So I'm going to give you a very simple hypothetical. I am a researcher who is interested in developing a therapeutic and I have scientific rationale that we're just going to accept for this, for people with C9 mutations who are slow progressors. How might I use ARC?
Well, you'd want to see, number one, how many folks are C9 carriers who are slow progressors. You'd be able to quickly define that, at least within this cohort. You could then look into the rest of that cohort's genome, right? To understand, are there any characteristics that correlate with that improved outcome? And does that genetic information, is it consistent with the hypothesis that you're testing? So that's one way. Another way might be to reach out directly to us after you've found that cohort and you found something interesting, and ask if we might have any cellular reagents from those people. We may have collected fibroblasts from those people and cultured them here, or reprogrammed those fibroblasts into induced pluripotent stem cells. And we may be able to share those cells with you so that you could actually develop assays to test your therapeutic approach. So there are lots of different ways one could delve into the data set to make use of that.
Awesome.
Did that answer it?
I think it did. Thank you. Nadia, I'm going to let you get your notes together on some of the questions that are coming up in the chat. And I'm going to ask a few in the meantime that came in with people's registrations. And there are a lot of questions, Fernando, around how this can be integrated with other data sets across the ALS community, how people can collaborate with ARC. So how does this study compare and contrast this with Everything ALS, Answer ALS, the registry, stuff like that, if you could?
Okay, so I'll first say that our big effort here was to be collaborative and to share as widely as we possibly could. So it was to break down barriers. There are a lot of great programs that people have contributed data to, people with ALS have contributed data to, and are continuing to do so, that could be integrated or compared, right? So I actually think the most important thing is less trying to get all of the data under a single proverbial roof, and more, the key is more about making sure all of the data are available to everybody. The reason I say that is that I think there's a lot of value in coming into our data set and asking a question and seeing something that might show you a trend, maybe a relationship between two genes, and then being able to go to a completely independent cohort from a different study and ask the same question and essentially have immediate replication, rather than the scenario where we have one giant cohort, we ask a question, and then we say this deserves more study, right? Having multiple distinct cohorts has value. Now, part of making that work means having the global unique identifier to make sure that we don't have duplicates, and that needs to be solved, and we'll work to solve that with other folks. But it's more about openness, easy access to the data for lots of people. Now, the question of other groups. So Answer ALS has been an amazing program where they've collected lots of genomes, more genomes than we have, probably more than 1,000. Theirs is different in that it doesn't have the degree of longitudinal data that we have. They've done some work with digital biomarkers. That's been good. It doesn't have the same amount of data associated with it, but it's another great program that does integrate omics with clinical presentation and with induced pluripotent stem cells. And they've had a pretty big impact in the academic world as well. The Everything ALS Radcliffe study is another really cool program that they've initiated over the past year or two, which definitely goes, it doesn't have, to my understanding, any of the biological omics elements. So genomics, proteomics, transcriptomics, et cetera, and sort of the blood sampling and cell biology work. But it does go very intensely in digital biomarkers, and they're doing amazing work there. So our program, I think, is the longest running, most longitudinal, and multi-dimensional of the programs right now. And I'm excited to be able to share it and to work with those other groups to really compare notes and see what's in common and what's a little bit different. There may be important signals in that.
Great, thank you. Nadia, you want to switch on and off here, and you can do a question that's come through in the chat, and I'll go back, and having seen some of the ones, as I know you did, that came in before, if you could choose a different topic, that would be great.
Sure. So there was a question that we know the data is updated daily, but is there a lag from the time that we collect data until it appears in the dashboard?
Yeah, but not much. Like, it might be a week. It's really updated pretty constantly. If you're a user, you'll probably see new data like every other day or so. That's actually an important thing to remember, because if you look at something today, it might be a little bit different tomorrow. So if that's very important to you as a researcher, you want to download and sort of timestamp your cohort and know why it might be different the next day. You want to keep track of that. But yeah, it's a pretty regular update.
Great. And we also...
Oh, sorry, Nadia, you go.
Oh, one more, and then I'll let you go, Carol. We also had some questions about what we're tracking within ARC. So are we tracking people's treatments and potential clinical trial participation, supplements, other things like exercise? Are all of those things being tracked?
Yeah, so things that we track. It's a really long list, and that's probably a whole different town hall to go through sort of the participant portal and all of what we're collecting. But yes, ALS functional rating scale, self-reported medications, self-reported supplements, clinical trials. We also ask a lot about a person's lifestyle before they had ALS. So yes, family history, but also their own exercise history, their own occupation history. Where have they lived and when did they live there? And all of those data, they're not all here available in graphical format, but they are available through Explore in table format. So for the informaticists out there, that's a deeper dive, and we can walk through that maybe one-on-one. But there is a massive collection of data behind this that folks have access to. I hope that answers it. We also implemented a new, like, how's your mood, right? Like, how does a participant feel today? Are you, cheery or neutral or less happy? And that might actually be important when relating to self-assessed ALS functional rating scale, things like that.
Fernando, we've gotten a few questions as you're talking about things that we're tracking about mRNA vaccines, particularly COVID exposure and illness. Could you talk about that?
Yeah, we did get, we sought and obtained IRB approval to share a COVID-related survey with our participants that asked questions about whether or not you thought you had COVID, whether or not you had diagnosed COVID, positive test, whether or not one was vaccinated and with which vaccine and when. So we have been collecting those data from our participants. We haven't had a chance to delve into those data yet, but it's worth doing. We did this at the prompting of a participant who asked for this, and we realized it was a really good idea, and so we implemented.
And is that available through this portal?
Not yet. Sorry.
Okay.
We'll make it so.
All right, great. Nadia.
So is there going to be data on anticipated rate of decline with this? Will we be able to predict somebody's progression potentially using data we see here?
I don't know how to do that. I hope somebody on this call will sign up for access to the data and build those types of models. Some of that work has been done. Ernest Frankel has done some work like that out of Ernest Frankel is a fantastic researcher in his group out of MIT. And he's done some of that work with Answer ALS data. Would love for folks to do similar types of work and maybe use the self-assessed FRS data that we have here to see how that models as well.
Fernando, are there any strings attached if somebody comes to a conclusion and publishes?
Just say that you got it from us. That's the string.
Awesome.
Just an acknowledgement, yeah.
And some people have been asking about participation in the ARC study. And I suppose they're asking too about the data commons globally. Are there barriers to participating in the ARC study as a person with ALS if you live outside of the United States? And are there any barriers to accessing data if you are outside of the United States?
A person with ALS can sign up for the ARC study no matter where they are, but they may not have access to all of the elements of the program. So we cannot collect blood samples everywhere in the world. Right now, we can collect blood samples from continental United States and Canada, but we're not collecting blood samples from outside. A participant can select the elements that they want to participate, they can opt out of anything. If a participant wants to join and contribute ALS functional rating scale only, they get to do that. If they want to join and fill out surveys about where they've lived their life and what their occupation was, and they qualify for the accelerometers, they can do that. It's sort of a la carte for the participants. We are mindful of participant burden.
Awesome. Thank you. And I know that there's at least one person on here who suggested they could send us their blood sample by DHL, which I really appreciate. But I imagine the IRB wouldn't love that too much until protocols were in place.
So it's the time that it takes to get the samples to us. That's a big part of it. So we've validated and have a strong understanding of how the samples hold up with a 24-hour FedEx in the 4-degree centigrade cold box. And we store all of those exactly the same way. We try to keep it as uniform as possible. When you start getting to, going across the pond, across the Atlantic or the Pacific, then the amount of time that they're in transit's higher, and that increases variability and our confidence in how we can compare the samples. So that's really the limitation there. I would love, I would love to have like satellite locations where we have folks there who can collect and process the samples on that continent so that they could be treated the same way, and then we could bring them together. That is a bigger vision for another time, but I'd love for that to be a thing that happens someday.
Nadia, you're up.
I have one more here. Are there any protections in place, Fernando, to make sure that nobody's using the data to harm people with ALS who are participants in the study?
The data use agreement does legally bind people to not make any attempts at all to re-identify. That's another string, right? Like, you're not allowed to try to re-identify any person, and therefore you wouldn't be able to harm any specific person. That's a main element. That's why there is a data use agreement, rather than just having this be something that's sourced on a website that people can just kind of go to willy-nilly. We do need that legal restriction. So we do try to limit that risk.
And a couple of people have asked about the NIH strategic plan and its call for a natural history study, and would love to hear you talk about how you think this might relate to that.
I think there's consensus in the research community that we need more ALS natural history study data, and that a lot of the elements that were noted in the strategic plan that would qualify kind of the perfect study are integrated here. The one thing that isn't quite there yet is the electronic health records, and I think we'll be there in the next couple of months. And so we would have checked off all of the boxes. The one beyond that is scope, number of participants, and we hope to be able to address that too. So I don't know if this is the study, but it certainly should be considered a study that can be integrated into that plan, right? We're sharing the data, making it available. It certainly goes a long way to getting to where the strategic plan wanted to go.
Awesome. All right, before I have one more thing I want to make sure we do, but before I do that, I want to be sure that there isn't anyone on the Zoom who's living with ALS and has difficulty typing in the chat and may want to use their voice or computer assistance to ask a question. I will give that a minute or so. If you're capable of raising your hand with your computer Zoom and anyone sees a raised hand, please let me know. And actually, Fernando, I can ask you this question while we wait and see. And if there is someone who does want to use their voice, great, or their computer, great, you can interrupt me. But Fernando, how do people access this information? What do they do next?
I'm horrible. I should have just had the link. I didn't add that to the slide. So if you go to als.net, click on the research drop-down menu, it'll take you to ARC or ARC Data Commons. You click on that, and then there's a straightforward button that gives you where you enter your name and any affiliation if you have one. And you would submit an inquiry, and Alan Primisari will, or another member of our clinical team, will reach out to you with the appropriate data use agreement for your type of organization or lack thereof. And then you work through, sign it. If you want some guidance on how to use it, he'll accommodate and he can make that happen. If he gets overwhelmed, maybe you'll get stuck with me. But hopefully this would be a good resource for you. Having watched this, I think we'll make this available, right? We recorded it, we can post it. And it might make it easy for folks to use. But that's it. Go to the website, click on the link, put in your name, contact information, and then we'll reach back out.
And everyone who is an academic researcher with a nonprofit or a clinic can expect to pay how many millions of dollars for this data?
$0.
Right. And
there is a maintenance fee for for-profit enterprises, but it's low. It's not designed to be a major barrier to entry. It's just to help us pay for the cloud computing costs.
Which some of you on this call, I may come back to you for help with that too, just FYI. But the most important thing here is that everyone can leverage this data quickly, efficiently, in honor of all of the people in our lives living with ALS. You know, I watched Fernando do a demo the other day. A couple of you were on that call. And there was something that struck his interest when we drilled down a little bit, and he wrote it down. And it struck me that, you know, we don't have enough people. You know, we have 30 scientists here whose eyes are on 35 terabytes of data. We need more eyes and brains on this in order to turn it into something that's going to help people as quickly as possible. So it's time to proudly share that and ask for everybody to jump in and leverage that. So feeling pretty proud about this.
Thank you, Carol. It looks like Michael Joyce has their hand up.
Oh, great. Michael, you're up.
I've had ALS since July of 2016. Been on Radicava since September of 2017. Participated in the Dartmouth study 2017, 2018, typical blood, nail clipping, skin, and et cetera in history. Was that all for naught? And I don't think the people who are following me are submitting any data concerning Radicava now that I've survived into 2023 with no bulbar symptoms.
So I can't speak to that specific program. I'm sure it wasn't all for naught. I'm sure there are folks who are using those data and analyzing those data. I can tell you this, building data sharing platforms like this one is hard. Like we worked hard we collected the data, and people have contributed data incredibly graciously to us for 8 years. And we had them, the data were all locked in, servers right here. It took a very concerted effort and a lot of man hours to build this thing, to move all the data into a format to where it could be used. So I'm sure there are folks who are accessing those and they're making use of it. There could probably be more use, because I know that there probably could have been more use for what we were collecting. But we've solved that now, and we hope to solve that going forward. And we're also happy to share with folks what we did. If they want to implement it as well, we can give some guidance there. I don't know if that answers your question, but thank you for contributing to research. It's incredibly important. Thank you to everybody who has contributed any time and effort to make this thing possible. We hope this is a force multiplier of that effort. that allows anybody who cares about inventing treatments and discovering biomarkers for ALS can use this now to move the needle. The data that you give today can be used forever and ever. And that's a really important thing. There's no expiration date for data that you've contributed. So thank you.
Awesome. I'd like to thank everybody as well for that and for being here and for everything you do as partners to us in all of our work together every day. For those who have questions that are unanswered, I think we have at least covered the major themes, but we will have a transcript of this chat and we will run through it. And one of those development directors, raise your hands again, will reach out to you and answer your question. We're looking for your feedback. Be honest with us. If there was something you didn't understand here, we want to know. It doesn't make us feel bad. Thank you again to our sponsors. And if I'm missing anything, somebody speak up. But otherwise, we can unmute everybody, and anybody who wants to chat for a couple of minutes, I'll stay on. And Fernando and John, I think we can probably unshare, right? Are we good, John? Great. All right. If anyone wants to stay on and talk about this a little bit, I'm happy to stay on. You're welcome to unmute yourselves. And thanks, everybody. Thank you, Fernando.
Thank you all. Great to see so many familiar and new faces. Appreciate it.